Hello, my name is Chris Bunker. I'm the Senior Director of Business Development at Advanced Cell Diagnostics, which is part of Bio-Techne Corporation. Today, I'm going to speak about our proprietary RNAscope and BaseScope in situ hybridization assay platforms and their use in morphology-based gene therapy, biodistribution, and transgene expression analysis. The phrase morphology-based biodistribution was coined by Asaf and Whiteley in their review article on considerations for preclinical safety of AAV gene therapy products. In their words, the addition of morphology-based methods such as in situ hybridization for assessment of AAV nucleic acids may be critical to understand the cellular distribution within a tissue and provide unique insight that can influence safety and efficacy considerations. For our part, Advanced Cell Diagnostics has tools that are uniquely able to provide this type of morphology-based analysis of gene therapy nucleic acids. As an organization, again, we are part of Bio-Techne. Our products are distributed worldwide, and we also have a pharma assay services CRO that can use our products in assays for you. We have nearly 25,000 probes in our catalog, and we can use these probes or develop new probes to detect essentially any RNA molecule in intact fixed tissues. So our technology is called RNAscope. However, it is able to detect both AAV DNA, genomic DNA, as well as transgene mRNA. In the context of the types of tissues that gene therapies are being applied, retinal tissues, liver, brain, in all cases, the viral nucleic acid that's being delivered and the transgene that is being expressed from that vector can be detected with probes either to the transgene sequence or its corresponding RNA, or probes to non-coding regions, promoter regions, or the antisense strand, which is also not transcribed. These assays provide us with a means of visual assessment on a single cell level in intact tissue of both vector copy DNA, as well as the transgene expression from those vectors. The RNAscope method has three key features. The first is that it's intended for and enables detection of RNA nucleic acids in formalin-fixed parapermitted tissues. It has the ability to detect and then quantify single molecules of RNA in fixed tissues, and it has very high level of specificity, much like any extraction-based nucleic acid assay. It's slide-based like IHC or immunofluorescence, but in this case, we're hybridizing with oligonucleotide probes, which then through a series of amplification steps will localize signal to the point where a visible dot will appear for each individual molecule being detected. A chromogenic dot in the case of HRP or alkaline phosphatase assays, or fluorescent dots in the case of multiplex fluorescent assays. The RNAscope probe design is such that signal is dependent on paired oligonucleotides hybridizing to the target sequence. A typical RNAscope probe will consist of a large pool, about 20 of these pairs, tiled along a region of about 1 kb of sequence. Each one of these pairs is able to generate signal. So by incorporating a large number of these oligo pairs into a given probe, we build in a high level of redundancy to ensure high efficiency of molecular detection. Every single one of these oligos is tested in silico to ensure that it will hybridize only to the intended target sequence and not cross-hybridize with any other sequence in the sample. We have two in situ hybridization technologies, RNAscope and BaseScope. Between these two, we can detect essentially any RNA molecule as well as any AAV vector in intact fixed tissue. RNAscope has a number of features that make it more suitable for detecting most endogenous transcripts, and it also allows for a higher level of multiplexing. BaseScope is an assay which has specific characteristics that enable for the differentiation between very highly homologous sequences, such as in the case of detecting a human transgene in the background of, say, mouse or even non-human primate tissue. Any situation where there's a very high level of homology, we would use BaseScope as compared to RNAscope, even to the point of being able to differentiate a wild type sequence from a point mutated sequence. In any case, starting from a gene of interest or a sequence of interest, we can design a probe with one assay or the other for in situ based detection of the intended target sequence. Both assays use the same signal amplification methodology where Paired oligonucleotides hybridizing to a target sequence, whether it be an AAV vector or transgene transcript, signal will be amplified when two oligos hybridize to the target sequence and then also simultaneously hybridize to a preamplification module, which will then hybridize to amplification oligonucleotides and then labeled oligos for signal detection. This amplification only occurs when multiple oligonucleotides are hybridizing at the same time in the same place, which is how we avoid any background in this assay. This signal amplification results in sufficient signal to see each molecule as an individual dot. That's illustrated in this data here from the first publication with the RNAscope method. In this case, we're showing an example of two experiments, one with HeLa cells and an RNAscope assay, in this case to detect the HER2 transcript, and in the other, a plate of HeLa cells where RNA was extracted and solution-based measurement of HER2 transcript was performed in the context of in vitro transcribe methods that could be used as a standard curve. In both cases, the number of copies per cell for HER2 is approximately the same, 14 dots per cell in the case of the RNAscope assay, and about 17 transcripts per cell from extracted RNA. And this is simply to make the point that an individual dot corresponds to an individual molecule being detected. Now, if this assay is run under slightly different conditions, which enable permeabilization of the sample and exposure of DNA, this same probe, which is detecting HER2 RNA here, reveals two dots in those same HeLa cells when we're now detecting DNA. So this is the same probe being used under conditions which expose genomic chromatin, and this assay is now showing two dots, which again emphasize the fact that a dot corresponds to a single molecule, whether it's an RNA molecule under an assay condition that's intended to detect RNA, or as shown here, the same probe detecting two genomic loci in HeLa cells, which are wild type for HER2, and multiple loci in the case of SKBR3, which is amplified for the HER2 locus. The next few slides will illustrate examples of the sensitivity achieved with RNAscope and how that compares to other methods like IHC and qPCR. We do like to emphasize the fact that a few dots matter most often, and that that's what we can detect with the RNAscope assay. In this example, this is an image of an RNAscope assay performed on a mouse lung, where the probe is detecting the RO1 expression in pulmonary vein endothelial cells. This was in relation to a CAR T-cell program, and it was this level of expression which, when these mice were treated with anti-RO1 CAR T-cells, resulted in pulmonary toxicity. And that correlated with infiltration of CAR T-cells as detected by RNAscope in the lower right, as well as corresponding activation of T-cells based on granzyme B expression. This was a safety assessment study which proved or demonstrated that a low-level expression of RO1 detectable by RNAscope and undetectable by IHC led to a phenotype that related to the activity of CAR T cells targeting RO1 expression. Another illustration of RNAscope sensitivity is shown here in relation to qPCR. This was for a clinical biomarker assay where NRG1 or heregulin had been identified as a marker that would predict patient response to an antibody therapeutic. Now, there were two factors that contributed to RNAscope being selected over qPCR. On the one hand, it was more sensitive when the sample was a very small needle biopsy. There was not sufficient material to get a reliable qPCR assay from that sample. However, because the RNAscope assay is measuring expression in individual cells, even from a small biopsy, there was sufficient expression information to be able to predict patient response. Moreover, it was the expression in tumor tissue which was most predictive of patient response, and that information could not be gathered from a qPCR assay, which would be a combination of expression that might be coming from tumor cells or stromal cells. And finally, in relation to NanoString here, we use the data shown here to just illustrate the additional information that comes from a visual analysis of nucleic acids. NanoString is an extremely sensitive assay suitable for RNA analysis in fixed tissue. However, it still provides an average value of expression across an entire tissue. And as illustrated here with these three examples, TNF-alpha, IL-1 beta, and CTLA-4, what we can see if we look visually is that there are very few cells that express these markers at fairly high levels, whereas the numbers shown at the top indicate that overall the expression throughout the entire tissue is extremely low. So the benefit of having a visual assay for these markers, or for instance for AAV vectors or AAV transgenes, being able to identify the number of cells that are expressing a marker and then measuring expression within those cells is the information that comes from in situ analysis as compared to extraction-based methodology. So now back to the primary focus here of methods for looking at AAV DNA and transgene expression. Now our assay is called RNAscope because it was originally developed prior to the development of gene therapy methods, and the intention was to detect low copy endogenous RNAs. What we've observed though is that probes to transgene sequences will of course detect the transgene mRNA. In addition, if we design a probe to a non-transcribed region, either a promoter region or if we target the negative or antisense strand, that probe will also detect the AAV vector DNA. So we have assays where we can incorporate probes for both transgene expression mRNA as well as vector DNA in the same assay. And these assays can be performed in any tissue to provide biodistribution analysis of vector genome as well as expression. And I'll use the next slides just to illustrate a few examples of that. So here's an example of an RNAscope Duplex assay where we did combine probes for vector, in this case the CBA promoter probe, chicken beta actin promoter probe, is detecting DNA signal only, and the GFP probe is detecting transcript. This is in non-human primate eye, retinal tissue, and you can see obviously very high level expression. This was local administration of the virus, so there's very high copy of both virus and transcript. A little bit higher magnification here, you can clearly see some nuclei which have very strong staining with the CBA promoter probe. That's of course again genomic DNA, and then other cells where you see both GFP signal for the transcript as well as the nuclear CBA signal. So the ability to identify which cells have vector and which cells have both vector and transgene is the kind of information which comes from a duplex assay like this. Here's another example where we're looking again at non-human primate eye, retinal tissue, with a probe to an AAV transgene for Cas9. The signal is only observed in the photoreceptor layer. Presumably that relates to tropism of the virus, but here again, the ability to see that expression in a specific layer is what we can provide with an in situ assay as compared to, say, extracting RNA and measuring total amount of Cas9 transcript or vector sequence from that sample. Here is another further example of non-human primate retina, where I presume the administration of the virus was into the intravitreal layer, and as a result, there's a large amount of vector signal outside of the cells themselves. In addition, there is, of course, also vector signal in red inside the cells, and a subset of those cells also express the transgene. In this case, we're using a probe to a codon-optimized human sequence as the transgene being detected. One more further example, again, a duplex assay. In this case, we're combining a probe for the AAV vector. The very few dots you see in the nuclei are vector signal. This was combined with a Rbfox3 or nu-N probe to identify neurons. In this particular region of the brain, there are a few cells that have low copy numbers of AAV vector. Those cells are predominantly or exclusively not neuronal cells. They do not co-stain with the Rbfox3 probe. In this instance, RNAscope was used to detect an AAV transgene in mouse brain. And again, Rbfox3 was used as a neuronal marker. In this instance, the expression of the transgene is predominantly in Rbfox3 positive cells and identifying neurons. And what we have here is data from a customer which shows very nicely two different applications, one being fluorescent detection on the left-hand side and the other being bright field chromogenic detection. Same assay, just different readouts, fluorescent or chromogenic. So you can clearly see very strong signal with the transgene probe. What you probably cannot see at this level of magnification is a very low level of signal in the PBS samples for the transgene. And this is because the transgene is a human sequence. The probe is to that sequence. However, there is cross-hybridization of the human probe to endogenous mouse ortholog sequence. And we would have known that, we would have told the customer that had we known what their intended purpose was. So this is just a context in which to reintroduce the BaseScope assay, which is what we do recommend to customers anytime there is a very high level of homology between any background sequences or off-target sequences in the intended target. Now we have a very nice interface on our website for new probe design, and if you engage with that interface to request a new probe, the first two questions are, what is the species of the target sequence, and what is the species of the tissue on which will be tested? And with that information, we can verify before we even make the probe whether or not we can design a specific probe with RNAscope, and if we cannot, then we will use BaseScope. So BaseScope has the characteristics of a much higher affinity for the target sequences. It uses more like locked nucleic acids for hybridization, and therefore we can differentiate much more highly homologous sequences. So this is the assay we would recommend in cases where there's high homology, such as for a human transgene in a non-human primate or even a mouse background. So just to illustrate that, this is also some customer data showing a BaseScope Duplex assay. Again, same combination as you've seen previously, AAV vector in blue and a transgene sequence, transgene signal in red. And you can obviously see much more signal for red and a few dots in the nuclei with blue. And that's charted in a histogram on the right-hand side. Now, these signals can be quantified. Any assay signal can be quantified. Our preferred software is Halo from Indica Labs, and this will enable us or you to quantify either transgene expression and or vector copy number in each individual cell within an intact tissue. So this allows for the collection of literally hundreds of thousands of data points for numbers of cells that are positive for vector genome, how many copies are present in each cell, and what level of expression is present there if you're using a transgene probe. As a transition to a related topic, I first want to re-emphasize the point that a probe to an AAV transgene will generate both RNA and DNA signal. Shown here is muscle-specific AAV transgene expression, where you can see in the three panels very clear RNA signal in the cardiac muscle and skeletal muscle on the right and left, and in the middle, clear nuclear DNA signal where the transgene is not being expressed. So the few dots you see in each individual cell in the liver correspond to AAV genome, and the signals you see in the cardiac muscle and skeletal muscle are predominantly a transcript. Of course, there'll be some few dots in the nuclei of those cells as well, corresponding to genome. But this again is just to emphasize the fact that these probes detect both RNA and DNA. RNAscope is also being put to very good use in lentiviral-based cell and gene therapies. I speak at much greater length about CAR T-cell and TCR T-cell applications in a different presentation. Here, I will just use a few slides to illustrate its use for detection of lentiviral DNA sequences. So much like the situation with AAV, where we can use probes to detect a transgene, as well as probes to an antisense strand or negative strand to detect the AAV vector, it turns out the same is true for lentiviruses. And this came as something of a surprise to us. Typically, or almost invariably, RNAscope probes do not generate any genomic signal. The probe to a endogenous mRNA does not generate signal corresponding to the genomic sequence encoding that RNA. But we observed, and others have observed, the fact that lentiviral sequences appear to be more accessible to probe hybridization. That may be how they choose their sites of integration in the first place, but we've observed that by designing a probe to the non-coding strand of a lentivirus, we can provide a tool and the means to detect cells where the lentivirus has been integrated. And this is illustrated in work from Jake Estes and others, which show the use of RNAscope for the detection of SIV. What's shown here are two assays, a duplex assay using one probe in red to the coding strand, which is detecting lytic SIV, and another probe to the non-coding strand corresponding to integrated viral DNA. And that can be highlighted here, shown here in circles. You see individual dots in cells where the virus is integrated, and the red signal is, of course, the much stronger lytic signal coming from the viral RNA. The ability to detect individual copies of a virus integrated into a host cell genome has made RNAscope a very useful tool in the context of HIV vaccine development, as well as vaccine development for other viral pathogens. Investigators, included Jake Estes and others, established and proved to themselves that they were able to detect a single copy of the SIV or HIV genome integrated into cells. And that work is shown here. In addition, they also verified the near perfect correlation between measurement of SIV genome copy number by RNAscope and measurement by qPCR. So these data also further support our assertion, which I made earlier in the presentation, about the fact that the RNAscope assay is detecting individual molecules of RNA, and that an individual dot corresponds to one molecule of RNA in the context of an RNA assay or a genomic DNA, as shown here. In the context of cell and gene therapy, this same assay can be used as a means of detecting and tracking the presence of lentiviral transduced cells. This is data generated by Bluebird Bio, looking at cells transduced with lentivirus and expressing a therapeutic transgene. What's shown on the left-hand side is a DNA-ish assay using RNAscope. In the middle, same RNAscope, different probe, now detecting the transgene expressed from that lentivirus. So in this case, Bluebird actually preferred to use the DNA-ish assay as a means of detecting and quantifying the numbers of cells present in the bone marrow which had been transduced with their lentiviral transgene. The same assay has been used and can be used in human bone marrow biopsies, shown here, similar assay, but in patients following treatment with a cell therapy where the cell therapy is being delivered by lentiviral vector and detecting transduced cells with a probe to detect the lentiviral DNA. So just to kind of segue very briefly to another method which ACD has developed for use in a DNAscope assay, which we will release later this year or early the following year. However, the method of RNA removal has become a very useful tool in the context of AAV gene therapy studies. I've mentioned previously the fact that our probes to assay a transgene will generate both RNA and DNA signal. By using these RNA removal methods, as shown here, we can completely remove all RNA from the sample. Most strikingly is on the very right-hand side, mallet1 is extremely high copy. It's also a nuclear transcript. It's a long non-coding RNA. And following RNA removal, that signal is completely removed. So by doing studies, say, with a transgene probe expressed off of AAV, plus and minus RNA removal provides us with a means of quantifying signal derived from the transgene as well as signal coming from the vector. To review, the data that I presented regarding the detection of AAV vectors and transgene expression in tissue-based assays are utilizing our RNAscope and BaseScope in situ hybridization assays. All of these are off-the-shelf kit-based products. We have, as I mentioned, tens of thousands of probes to detect endogenous sequences, and we've designed hundreds of probes for detecting various different AAV vectors, as well as lentiviral vectors and their transgenes. The RNAscope assay is our primary assay. That's our default assay. It has a number of features that make it somewhat more useful in certain circumstances. In particular, it allows for higher plex fluorescent assays, 3 and 4 plex fluorescent assays, which allow for inclusion of cell markers as well as detection of transgenes and or vector sequences. The BaseScope assay is what we would utilize in instances where the target sequence is too highly homologous to other sequences, whether it's other related homologs or when trying to differentiate a human transgene from ortholog sequences, whether they be mouse or non-human primate. In addition, the BaseScope assay is able to target very short sequences. So in instances where AAV gene therapy is delivering, say, Cas9 in a guide RNA, the BaseScope assay can be utilized to detect that single guide RNA. Now, neither one of these assays is suitable for microRNA detection, which in some instances is being delivered through an AAV vector. However, a soon-to-be-released microRNAscope assay will be applicable for detection of microRNAs, siRNAs, and ASOs. So again, all of these are product-based kits which can be purchased for use in your laboratory. Alternatively, you can utilize our pharma assay services. We have a CRO, which will be GLP compliant by middle of 2020. This lab does next to nothing else other than run in situ hybridization assays. They do also run some IHC assays, but we run nearly 20,000 slides a year of RNAscope and BaseScope assays, as well as now microRNAscope assays. These assays are being run fully automated on 14 Leica and Ventana autostainers. We have, of course, slide scanning capabilities. We can perform image analysis with Halo software, and we also have board certified pathologists on staff. And finally, our emphasis with RNAscope is the ability to perform morphology-based analysis of AAV vector, biodistribution, and its gene expression patterns. We can do this with probes for essentially any target transcript, any transgene, any vector. We can easily design probes that will differentiate from either codon-optimized or normal human sequences against a background of mouse or non-human primate orthologs. We can multiplex these assays together vector transgene, as well as with any number of cell markers. And ultimately, the readouts are showing you detection of individual molecules of either vector, genome, or mRNA, which can be quantified with image analysis software. I hope this information will be of interest and of value to your research, and that we will hear from you in the near term. Thank you.